The next speaker is Limin Hu from ICN2, and the title of his talk is Monitoring Generic Escherichia coli based on lateral flow biosensor for water fecal pollution assessment. Thank you. The floor is yours.
Okay, thank you, ICN2, for organizing this interesting workshop. Okay, today I'm glad to just introduce a calorimetric lateral flow biosensor applied to monitor generic E. coli for the fecal pollution assessment. So first, as we all know, the water is really the most important ingredient of life. And currently, the water fecal pollution just threatens water quality worldwide and also has direct detrimental effect on human health and also global economy. Fecal pollution, fecal bacteria, especially the E. coli, is the best indicator for water microbial pollution, just due to two reasons. So first, because E. coli is the most abundant bacteria in mammal digestive system. And second, just E. coli is more easily detected compared than other water-borne pathogens. And for the normal gold standard technique for the E. coli detection is bacterial culturing, like some membrane filtrations, and also some DNA-based techniques, for example, the qPCR. And these standard techniques still have some disadvantages. For example, time-consuming, and also it's complicated, sometimes and also relatively expensive. In addition, they also need some well-trained personnel and also highly equipped laboratories. So our purpose just currently, actually, there are already existing some cheaper and also convenient techniques. In all of these alternative techniques, lateral flow strips is one of most outstanding approaches. And here is the structure of the lateral flow strips. Actually, I think a lot of people already know this product, because in this pandemic, a lot of commercials lateral flow strips already just applied in detection of antibodies of SARS-CoV-2. So this strips is just a paper-based biosensor. Here you can. This is have various pads just assembled, and just based on the capillary functions of the absorbent part. And then when we just loaded the samples on the sample pad, the labeled conjugations will just flow and captured on the detection part. And then for the naked eye, or just using some calorimetric readers, we can just get the signal. And in these biosensors, the labels, or namely the transducers, is one of the key elements. And the nanoparticles is mostly selected as the labels, just because it's really easy and versatile synthesize. And also this is stable, have the long shelf life. And this is have the very intense red color. You can directly use naked eye, or also you can using the calorimetric reader to qualitative or quantitative just detection of the analyte. And most importantly, these nanoparticles can easily conjugations to different molecules, for example, the antibodies, DNA, or optimal texture. Although they already have some nanoparticle-based lateral flow strips to detect the E. coli, but they still have some disadvantages. Like first, they need some additional steps and also long processing time to get a greater get better sensitivity and lower LOD. And these products normally just applied in the food samples detection, but few of them do it for the water analysis. Most of these just detection certain E. coli strains using monoclonal antibodies. This is just would thus neglect the overall water microbial pollution. So for us, our purpose just developing gold nanoparticle polyclonal antibody-based lateral flow strips with an easily portable calorimetric reader to detect all strains of E. coli species. So in this work, first we just developed a novel characterization method of lateral flow strips materials using the bioluminescent bacteria Allivibrio fischeri. So this one is because this bacteria, this bioluminescent bacteria has a similar shape and sense to the E. coli. So we can directly using this bacteria to mimic the dynamic flow of the E. coli on the lateral flow strips. So we just culture this Allivibrio fischeri in the biolab, and when the bacteria can just produce the bioluminescence, we dispense this bacteria on the sample part, and under the dark environment, we can using the smartphone to record the bioluminescence like every 50 seconds and until 10 minutes. So until an optimal lateral flow strips must allow most of the bacteria cells, just thus the bioluminescence to reach the absorbent part. Here is the real experiment testing the flow dynamics of different nitrocellulose membranes as detection paths in lateral flow strips using this Allivibrio fischeri as the flow indicator of the 10 minutes. From here, you can see for this one, we at least 17% of the bacteria flow throughout the detection flow. So in the next step, we can directly discard the C95 and the HF114. And under the optimal parameters, we just make the lateral flow strips. So the left one is just the commercial portable calorimetric reader, and left is the real test strips. From left to right, just corresponding to the increasing concentration of E. coli. And in this project, we also just combination very simple filtration system to just want to improve the detection, the sensitivity. Here we just concentrating the 100 times, and from here we can see when we just connected to the filtration unit, the sensitivity just improve the 2 orders magnitude. And we want to prove the universal of university of these biosensors. We tried through different kinds of the E. coli species, and finally just proved all of these can just reaches the limited of detection of 10 to 6 CFU per milliliter. but this one is without the filtration. And we also tried to prove this biosensor's selectivity. And from here, we also tried the others bacteria species like Salmonella and also Vibrio fischeri. From these results, we can see this little flow strips not only can detect all of the species of E. coli and also can discriminate other species, for example, the Salmonella and Vibrio fischeri, just prove that this have a very good sensitivity and also have the good universality. We also tried just applied these strips in the different sources of the water samples and through suspected the E. coli in different kinds of samples, just to prove this this method is suitable for the real samples detection. And in the real samples detections, when we tried the different pre-concentrated factors like 20, 100, 200, and 500 times concentrated, and see we just found when we concentrated 500 times, we can reach the limited detection of these strips. So from here, just to prove the is can realize the real samples detection. So this is conclusions. Actually, I already introduced all of these parts just in the every slides. So finally, I will thank because this project just funded by the Horizon 2020 Engage, and I acknowledge all of the colleagues involved in this project. Okay, thank you.
Thank you very much for this interesting talk. There is time for questions. No questions, so we can move to the next. Thank you.
Okay, thank you.
Speak again.
Thank you.